Good morning everyone. Today in this video I'll be talking about the DNA polymerase 1 fragments which should be used in Sanger sequencing method. Okay, so DNA polymerase 1, if we performed its proteolytic cleavage, okay, proteolytic cleavage is performed in case of DNA polymerase, it will give rise to fragments or two components. One that is known as large C-terminal end. Okay, the second one is small N-terminal end or N-terminal fragment. So one is large C-terminal fragment, one is small N-terminal fragment. Now this DNA polymerase have three functions. One is obviously polymerase function, one is 5' to 3' exonuclease activity, and another one is 3' to 5' exonuclease activity. Now this large C-terminal end that has only polymerase activity and 3' to 5' exonuclease activity. Okay, and in case of this N-terminal end, it only has 5' to 3' exonuclease activity. Clear? So this fragment, this large C-terminal fragment which has two different types of activity is known as Klenow fragment. I hope you guys will heard about it in your colleges. So what is Klenow fragment? So this is the Klenow fragment which has both polymerase and 3' to 5' exonuclease activity, and the small N-terminal fragment that has only 5' to 3' exonuclease activity. So if we want to get same 5' end containing DNA which will be produced, that is the complementary copies of the single-stranded DNA should be sequenced from the bacteria. Okay, we need this Klenow fragment only because they has only the 3' to 5' exonuclease activity. So after polymerization, 3' to 5' to exonuclease activity will cut from the 3' end. Okay, so if we just cut it down, this portion from the whole enzyme, we will get our desired same 5' sections of the complementary copies